Okay, so you can go to next slide. Yeah. So there is. I will give you a brief introduction about bacteria and swimming activity of bacteria, and then I will I have two parts in my talk. First part will be about filtration of bacteria in porous media, and then I will do also I will talk about when there is collective effect in the suspension and the effect on flow in porous media. So if you go to the next, so yeah, next. Yeah, okay. So next one. Oh yeah, okay. So very good. So bacteria, they use flagella to move. So yeah, if you can show all the yes, perfect. So you see bacteria, they to move in the fluid, to swim in the fluid, they use flagella. So flagella, it's look like an helix, and it's made of proteins, and so the helix can be left or right-handed, and all these those helix are all over the bacteria, so they can be everywhere over the bacteria. So it's called peritriche. So it's like E. coli, and I will mostly focus on that type of bacteria. But you can have also bacteria that have only one flagella or flagella located at different place over the body of the bacteria. So there is like 5 to 6 flagellas, and they are connected to the body of the bacteria by nanomotors. So nanomotors, they can make the flagella rotate. And for E. coli, for example, when all the flagella, all the motors rotate in the same direction, you will create a bundle of flagellas, and you will see the motion of the bacteria. And that's on the next slide. Yeah. You see on the movie, you see some E. coli moving in a liquid. So when all the motors rotate in the same direction, you have a bundle, and you see that the bacteria is moving. So it moves at a velocity of about 20 microns per second. And after some time, you see you see the flagella, they unbundled. And this is due to the fact that one of the motors start to rotate in the other direction. So instead of clockwise, you have anti-clockwise rotation, and that creates the bundle. So here you have some typical size. So a bacteria is about 10 microns. It moves at a velocity of 20 microns per second, and a run duration, it's about 1 second. And so we can see in the next slide that when a bacteria moves in a liquid, it also displaces the fluid around its body. And it's what you can see on that image. You can see that you can see the flow lines of the liquid around in the vicinity of a bacteria. So you can see that the bacteria push the liquid ahead. So you have a flow from the head to the liquid, and also a push on the liquid. So by conservation, the fluid has to come back, and it's why you have these flow lines that come back to the on the bacteria by the side of the bacteria. So you can model that object by a force dipole, as you can see, and that will be key in my presentation. So it's very important that you understand that when a bacteria swim, it also displaces liquids around it. And that you can see on the next slide. The consequence of that flow field is that it will introduce hydrodynamic forces in the problem, and those forces, they will, for example, um, uh, be attractive when a bacteria swim very close to a surface. So you will have a kind of pumping effect. And so when the bacteria swim very close to a surface, you have this attractive force induced by the hydrodynamic forces. This has consequence, as you will see. And also when you have two bacteria, and so when they start to be close to each other, because of this hydrodynamic effect, they will start to swim in the same direction. And this will have important effect on collective motions. That will create collective motions in the liquid. So next slide. So I will now talk about filtration of bacteria. And on the next slide, you can see that bacteria, they can be are not only active because they swim. They are also active, chemically active, because they have they can produce some enzyme. An enzyme can help to trigger some chemical reaction. And it's this type of effect is used nowadays, for example, to create a skin on some plastic materials, or also is used to stabilize the grounds or to close some fractures. So I will, you will see on the next slide one of the use of bacteria. It's for soil reinforcement. So there is a company that is using bacteria to reinforce soil. So the idea, it's a special bacteria. It's a Sporosacina pastori. And this bacteria has an enzyme that is able to reduce urea into ammonium ions and carbonate ions. And the idea of the process is to combine these carbonate ions with some calcium ions to create calcium carbonate. Okay? And the idea is to use these crystals to bound grains between them. So you see on the next slide, on the next slide, you see the process. So you imagine you have a sands, for example, on consolidate materials. So you draw a hole, and then you inject bacteria. Okay, so you inject in the ground the suspension of bacteria, and then you inject urea, a bit of chloride calcium, and then after some times, you see that between sand grains, you will have crystals of calcium carbonate. And these crystals, they will bridge the grains, and this will reinforce the soil. So on the next slide, you will see that one issue with this process is to know where bacteria will be filtrate, and where the bacteria will be on the grains as function of the flow rate, as function of the chemical conditions. So it's because they want to optimize the process. They want to reduce the cost, so they have you can show some, yes. So they want to know where the bacteria will be filtrate. So to do that, so I will just briefly say a few words about filtration. That's on the next slide. So the basic idea, it's you have a certain quantity of bacteria, B, and you want to know how much bacteria will be on the grain. So that's the quantity S. And this process can be modeled by an absorption rate. So it's a very simple model. So we have a certain amount of bacteria that will leave the fluids and be retained on the grain. And so the profile of bacteria will be like an exponential profile with a typical length scale, which is related to the absorption rate and the flow velocity. And the quantity of trapped bacteria will be related to the number of bacteria and will increase with time, okay? So there is two ways to determine, yes, you can move next, yes. So there is two ways to determine this absorption rate. The classical way is to use a core and you inject a certain amount of bacteria at the inlet and you measure the quantity of bacteria at the outlet, okay? You can change the flow rate, you can change the size of the sample, and the absorption rate will be related to the difference of bacteria at the inlet and at the outlet. That's the classical method. The method we developed is based on a microfluidic, and the idea is to use microfluidic to measure as function of time the number of absorbed bacteria. So this is the quantity S, and we try to measure S as function of time, and the ratio, the rate of increase of the number of trapped bacteria is related to the absorption rate, K, that we want to determine. So on the next slide, you see it's just the microfluidic chip that we developed, and I will go. There is not much to say about that, but on the next slide, you see examples of experiments where we were able to measure the number of bacteria on the grain by image analysis. And you can see on the bottom, you see an image also of the calcite crystal after mineralization. So you see it's and the bacteria is inside the crystal, and the bacteria is trapped inside the crystals. So on the next, that's a comparison between our microfluidic method and the classical core measurement technique. So here you see it's the absorption rate that we measure as function of the ionic forces, or the amount of salt of ions in the solutions. So you see you have a linear dependence of the absorption rate with the amount of salt in the solutions. So this tell us that it's mainly because of van der Waals effect that the bacteria bound to the grain. And you see also that we have a very good agreement between the microfluidic technique and the measurements on cores. So on the next, you see visualization of flow the bacteria at the scale, at the pore scale. And one of the questions we had at that time was to see or to understand if the swimming activity will help to improve the process. Because the bacteria, the Sporosarcina bacteria is not motile. So the issue was to understand if those bacteria were motile, will this help the process? So to understand that, we developed another setup, which we try to reproduce on microfluidic chip the geometry of porous media. So it's like a rock. We wanted to do a kind of rock on a chip. So here you see so the type of chip we obtain. On the next slide, yes, you see a bit more. You see the microfluidic chip we use. So at the bottom right, that's the field of view we have with our microscope. So you see the circles are the grains, are the grains, and you can see some disorders, and you see the typical size of the grains is about 20 microns. On the field of view, it's 1 millimeter by 500 micrometers. So, and since we wanted to see the effect over long distance, we used a serpentine-like porous media. So we use that to increase the distances. So let's see what happened. So that's the microscope, the pumps on the flow cell. So on the next, you can see the typical flow field we were able to reproduce. So the dark blue, it's where you have grains. So the grains are in black blue. The light blue, it's where the flow velocity is very low, so it's like pores. And you see you have channels of high velocity. So when it's red, it's where the velocity is very high. And so you see these long channels where the velocity is high. So we have channeling of the flow like it's usually observed in porous media. So we now inject bacteria, and that's on the next slide. So we inject bacteria. We have a special software to track the bacteria. So we can obtain the trajectories of the bacteria. And what I will, so you see on the next slide, what I will compare now, it's motile E. coli bacteria trajectories. I will compare those trajectories with the trajectories observed with non-motile bacteria. Okay, and you see on the two pictures examples of trajectories that we measured. And so I will now compare the properties of those trajectories. So the first things we measured, it's the dispersion. So we said, okay, after a given time, what is the distance along the flow direction covered by the bacteria. So, and we made the distribution of those distances. So it's what you can see on the top figure. So, so the distribution is shown in blue, red, and yellow. And you see the difference between the three is the time that we left the bacteria moving in the flow. And you can see that for non-motile bacteria, the peak, the maximum of the distribution moves with time. So you have a convection of the population of non-motile bacteria. And in the same time, you see you have the spreading of the bacteria. Okay, and you see the dot line, it's an adjustment by Gaussian distribution. So it's quite classical. You see you have the peak that moves with the flow, and you have a dispersion of the population around this average position. Now, if the bacteria are motile, that's the bottom figure, you see that the peak, the maximum of the distribution, stay very close to zero. Okay, so this means that we have a retention effect. This means that a large quantity of bacteria does not move with the flow. Okay, and that's to understand where this retention come from. That's on the next slide. We can have a look on at the pore scale of the trajectories of some of bacteria. I don't know if you see quite well the movies, but the black, it's the grains, okay? You see you have regions where, the bacteria are in white. It's the white particles because the bacteria we use are fluorescent. Okay, so you can see each bacteria. So you clearly see that you have channels of high velocity, okay, where it's difficult to see individual bacteria, but and then you see that the bacteria, they are able to leave the flow, okay, then they go inside some pores where the flow is low, and then they move on the surface of the grains, and they move along the grains, and eventually they go back to the flow. So you see a retention effect comes from an exchange between moving particles in the moving fluids and the pores, and also the interaction between the bacteria and the surface is also an important factor. So we tried, that's examples of trajectories we can record, and you see that from time to time the bacteria leaves the channels and goes on the surface of the grains and stay for a long time on the grains and move between grains, and then eventually goes back to the flow. So on the next, you see, so that's the distribution of the velocities of the bacteria. So for non-motile bacteria, that's on the left, you see that the normalized distribution does not depends on the flow rate, and you can adjust this distribution by a gamma function. Now you see with motile bacteria, you have two changes. You see you have more bacteria um, moving with a low velocity and less bacteria with a high velocity. Okay? So on the next slide, you see we try to model this exchange between high flow velocity and low flow domains by so we try to upscale our observations from microfluidic scale to mesoscopic scale and macroscopic scale, and we use a CTRW method to bridge the different scales. And we were able to model this process by using a two type of domains. One domains where the fluid is moving, and some part of the fluid where you have no motion. And the exchange between the two is you have to introduce a trapping rate. You have a trapping time, the time that the bacteria will stay in the stagnant fluid. And so you have an exchange between the two. And then from those measurements, we are able to obtain also the macroscopic scale transport parameters, which is a retardation factor. And we are also able to predict the diffusion coefficient of the population of bacteria. So on the next slide, so it's a test of the model. So the symbols are experimental data, and the line are the model. And you see the model is quite good and is able to fit our data. And from the fit by the model, we are able to obtain the asymptotic transport parameters. So that's, and you see that you have, that's so we call that active filtration, because the swimming activity of the bacteria is taken into account through the two parameters, the absorption rate and the trapping rate. And here you can see the retardation factor as function of the flow velocity. So that's on the right. And you see at low flow rate, you have a strong retardation. So the bacteria, they are mostly on the grains or inside the pores. And when you increase the flow rate, you have less and less bacteria inside the pores, and they are more and more inside the flow. Okay. Yeah, so on the next, so I will move to the next part of the talk, and it's about the collective effect. So far, I talk only about bacteria, single bacteria. But what happens when you increase the volume fraction of bacteria in your suspension? What you observe if you have a rheometer, it's you see that when you have a small amount of bacteria, this is the black squares, you see that the viscosity of the suspension is equal to the viscosity of the suspending fluid. But if you increase the quantity of bacteria, so you increase the volume fraction in the suspension, you see that now you have a decrease of the viscosity. And this is very counterintuitive because Einstein proved many years ago that if you put particles in a fluid, you increase its viscosity. And it's not what we observed here. You see that if you increase the number of bacteria, you decrease the viscosity of the suspension. So on the next slide, you see how this viscosity decrease with the volume fraction of bacteria. So you see it's more or less a linear decrease until a volume fraction of about 0.8. And then after we have reached this volume fraction, the viscosity of the liquid is 0. Okay? And we were able to show that this is related to the occurrence of collective motions inside the liquid. And it's what you can see on the movie. You see you see on the movie, it's a fluid at rest containing 1% of bacteria. And you see that for that concentration, you see occurrence of what we call active turbulence. So you have collective motions, and there is a relation between these active motions and the fact that the viscosity becomes 0. So on the next slide, you see what we wanted to understand, to prove, or to test if you have bacteria, you decrease the viscosity. So what we thought it's that maybe we can use this suspension of bacteria and to have some viscous instabilities, because we know that when we inject a less viscous fluid into a more viscous fluid, then we will have Safman-Taylor instability or viscous instabilities, viscous fingers. And so we tried to see if we are able to observe viscous fingers with bacteria. And so that's on the next slide. Yes. So we developed an experimental setup. It's more or less LHO cell. So you see it's something 20 centimeters long, and the gap of the LHO cell is 0.5 millimeter. And what you will see, it's not single bacteria, but it's a population of fluorescent bacteria, and we can relate the local concentration in volume fraction of bacteria to the light emitted by the liquid. So it's on the next slide. Yeah. So you see a typical experiments where we inject the bacteria. So it's a low concentration of bacteria, and you see you have at the end you reach everywhere 1. This means that you have bacteria everywhere. And in the beginning, so maybe you can see one more. Yeah. So in the beginning, it's 0 because there is no bacteria. And then you see the mixing front, moving inside the flow cell. And in the next, so it's So now we can translate concentration into colors, and that's the field of concentration that we observe from the top. We observe from the top, and you see the bacteria entering into the flow cell, and you see that the front between the two, the liquids without bacteria in blue and with bacteria in red is stable. And you have a mixing front between the two liquids, and the front is stable and moving at the velocity of the average velocity of the fluid. Now, if you increase, so that's the next slide. If you increase the quantity, the amount of bacteria in your suspension, you see that now you have a finger that develop, you see, and it's so it seems that it's an instability that we are able to observe. So you see the finger. Yeah, you see the progression of the finger inside the LHO cell. And in the next, it's we done the same experiments, but we swap the two liquids. Okay, now it's blue, so it's a liquid without bacteria pushing a fluid with bacteria. And now you see it's a stable displacement. So it's very nice. So it's now it's stable. And we also done some experiments, I think on the next slide. So here it's a different flow geometry. It's a quarter-5 spot geometry. So we inject in one corner of the LHO cell, and we throw the liquid at the opposite corner. So you see on the left, when the quantity of bacteria is low, the front is stable, but when you have a large quantity of bacteria, the front is now unstable. And yes. So here, it's so you see we are able to observe fingers, and it seems that it's really very close to what we observe in the classical Safman-Taylor instability. It's not exactly the case with bacteria, because you see that we, in fact, observed fingers only if the amount of bacteria is above this zero viscosity limit. So it's a bit different because in classical Safman-Taylor instability, what you need is just a liquid with a less viscous liquid. If you have a less viscous liquid pushing a more viscous liquid, then you will observe an instability. For active Safman-Taylor, it seems that what you need a certain amount of bacteria, so you need to reach a certain threshold to observe the instability. It's what our experiment give us. So it's a bit different. So I will give a brief conclusion of my presentation. Just to say first, microfluidic is really a good tool to study bacteria, because we can control the environment, we can control the chemical environment, and also the geometry of the flow cell. Um, so swimming bacteria, they like to stay on the surface and they like to stay on pores. And it's why we have a strong retention effect when we use swimming bacteria. And this is fully due to the hydrodynamic coupling effect. And I also show you that you can control the viscosity of liquids using bacteria. And it's maybe something we can use as a future tool to inject things into porous media. So here it's you see that you can use bacteria to have a kind of active fluids with some special properties. And on the next slide, it's just some open questions. I think one open question is still the effect of flow, pore structures, and chemotaxis. So what is chemotaxis? It's just the fact that bacteria can sense the chemical environment and they can move towards a source of attractants. And this is used in nature in many, many different cases. And one situation is, for example, the colonization of roots of plants by bacteria. In that case, they use chemical signals to attract one type of bacteria. But so the issue is to understand the combination of flow, pores, and chemotaxis on those effects. So I will just finish by two last slides. So next slide is no, next slide. Yeah, it's all the people that participate to this project. So my colleagues, Karine Douarche, Frédéric Moisy, Mojtaba Jarahi, and Veronica D'Angelo, some PhD students, Julien Bouvard, Akash Ganesh, Adam Akrepi, and some colleagues from biology, Peter Margert. Simulations were done by Marco Dens, and also I like very much to work with Eric Clément and to have discussion with him. And I will just have a last announcement. So we have a co-fund project in physics in my university. So if you have a project on bacteria, porous media that you would like to propose, we would like we are open and we can exchange about possibilities in my university. So thank you very much for your attention. I hope it was not too long.
Thanks, everyone. Do we have questions